limitations that exist there, and help you better appreciate the technology that what's happening now and where things are going. So I like to start with this quote. This is from New York Times science editor Cordelia Dean. She wrote a book, I guess about 4 or 5 years ago now, called Making Sense of Science. And she writes that we live in a world in which researchers gather data, politicians, business executives, or activists spin it, journalists misinterpret it or hype it, and the rest of us don't get it. Whoever has the most money, the juiciest allegations, or the most outrageous claims speaks with the loudest voice. The internet, newspapers, and airwaves, the public discourse generally are all too often brimming with junk science, corrupt science, pseudoscience, or non-science. So I hope that as we go through these things, you'll appreciate where some of the great science is that's in forensic DNA, and where some of the things where people are pushing. I'll finish with some things about genetic genealogy and what's being said. There was a recent article in from last week, actually, in the Atlantic that talks about these very issues. So I'd like to begin with there's some great resources. If you're not familiar with this is there's an organization in England called Making Sense About Science, and they write all kinds of interesting articles. But this is one they partner with the European Forensic Genetics Network of Excellence, and he wrote a book called, or a little pamphlet, it's 40 pages long, called Making Sense of Forensic Genetics. And it's written on purpose to be available and accessible to the non-scientists as well as the scientists. And the final point they make in this is that as DNA profiling continues to grow more sensitive and is used in more investigations, the need for accurate communication between scientists and non-scientists only grows, both to ensure that their expectations of the technology are realistic and its limits are properly understood. So that's why I titled my talk, The Science of Forensic DNA, the Capabilities and the Limitations, because there are limitations to any technique, and we want to make sure we're using it properly. So this document has been translated into Spanish, into Portuguese, into Polish, through funding from the International Society for Forensic Genetics, and hopefully into other languages as well. Well, I enjoy studying forensic science and DNA, and I've had a chance to work in a number of places. I worked at the FBI laboratory to do my Ph.D. research. I worked at the Armed Forces DNA Identification Lab while I was a postdoc, and then at Nist. And then I worked for a startup company for a while in California, and then came back to Nist after doing my postdoc in the mid-90s, came back at the end of, and have been there for almost 22 or 23 years now. Working, right now I'm in the Special Programs Office at Nist, but prior to that I ran the Applied Genetics Group and did the research of new DNA technology methods. So I loved performing research in this area, writing articles and teaching others. So that's why I'm glad to have a chance to talk to you today. I also have a website, the starbase.nist.gov. It gives you all kinds of information on STR markers. If you're not familiar with it, that might be helpful. And then, as Mickey mentioned, I've written a few textbooks that are used all over the world and been translated into multiple languages as well to help with teaching about DNA testing. I did a YouTube interview that was published last October. I'll show the full thing at the end, just a link to it. But I'll provide these slides if you'd like, and Mickey as well, so that you can give them to people if they want to click on the links and so on, to be able to follow things. DNA is famous, of course, because of James Watson and Francis Crick first discovered the double helix in 1953, or described it, with data from Rosalind Franklin and others who had provided input to that. I had the opportunity a couple of years ago to meet James Watson. He was 90 years old at the time, spoke at a conference, and gave him a copy of my latest book. And it was a neat opportunity just to meet him. If you look on my spines in my book, people that even have them probably haven't noticed carefully, but I just like the Da Vinci Code, I actually put some clues in there. I call it the Butler Code, that's put into the last 3 books. So if you haven't noticed that before, if you have one of my books, you might try to investigate and interpret that. You have to, there's some cool things that I've tried to build into it like that. A puzzle, I always like solving puzzles. So I work at Nist, the National Institute of Standards and Technology. If you're not familiar with us, we were originally called the National Bureau of Standards. The roots go back to the Constitution, where there has to be a place to be able to make sure that we have the weights and measures measured properly so that we can then transfer information and particularly with commerce, that things are weighed the proper amount or measured the proper length and so on. And when the additional computers came in the 80s, we added the technology part. Congress added that to say that we would also be in charge of computer standards. And so our primary campus is in Gaithersburg, Maryland, which is where I live, just outside of Washington, D.C. We're part of the U.S. Department of Commerce, again, with the Patent Office and other organizations to help make sure that commerce happens well. We have more than 3,000 federal employees, and we have a number of associates, people that come and work with us. We make reference materials, and we also define time. The official time.gov website is run by Nist. And so in terms of DNA, one of the reference materials we supply helps calibrate laboratory measurements and help with the traceability back to a common standard so that labs all over the world can compare their results because they're traceable to that standard. And if anyone's interested in doing a postdoc, that's how I got started at Nist back in 1995, was a postdoc. It didn't pay that much at the time, but it's about $65,000 or $70,000 now that's paid in this postdoc. And so if you're interested in that, it's very competitive, but it's a great opportunity to come work with some great scientists. So in our forensic science efforts at Nist, we have three primary pillars: research, standards, and foundations. So under research, we do research in biometrics and DNA and digital evidence and statistics, drugs and toxins, firearms and tool marks, and trace evidence. So we have research teams that work on this and collaborate with people all over the country and all over the world. We also fund a center of excellence called CSAFE, which is the Center for Statistics and Applications in Forensic Evidence. And that's headquarters out of Iowa State, and there's a number of partner universities that work with them. And then on the standard side, we work with the Organization of Scientific Area Committees to help with standards development, to help strengthen forensic science through how detailing the standards that will be used. And we have a lot of other efforts that go along with this, helping with evidence management, the human factors involved with handwriting or with fingerprints. We publish reports on that. There's an effort going on with DNA analysis and interpretation, the human factors efforts there. And part of that comes down to designing process maps. What are the processes that are going to go through with each of the forensic disciplines? And we're in the process of doing a number of those process maps right now. And then what I'm really focusing on right now, and I head this effort at Nist right now, is doing the foundation reviews. So we've been tasked by Congress to look into what's the underpinning knowledge, the foundational knowledge of each of the forensic disciplines and the various methods and practices that are involved. And so we have efforts in DNA mixture interpretation right now, bite mark analysis, digital evidence, and firearm examination. And then finally, we communicate our results with the community, and we have DNA explainers. There'll be other explainers like that that'll be designed for other disciplines to help with explaining complex issues. And then we disseminate press stories. Rich Press in our public affairs office writes stories, and then we share those with the community. So that's just a little bit of background about where I work. So I want to get into a little bit of background about DNA, and then we'll go into some more of the test methods and so on that are used, and then finish up with some of the practical applications what are being faced today. So DNA can be biological evidence. It can be used to obtain DNA from blood, semen, saliva, urine, and other tissues that, as you see noted there. And originally we had to have a large amount of DNA, about the size of a dime, when DNA testing was first developed in the late 80s. But now we can do testing with that you don't even see the DNA. The swabs only have a few cells. So ideally, we like to have more than 100 cells, but DNA profiles can be recovered from as little as a single cell in some cases. And that leads to some other challenges, which we'll talk about a little bit later. So our human genome contains both nuclear DNA, which is where the control center of the cell is, and mitochondrial DNA, the powerhouses for the cell. There's actually hundreds of copies of mitochondrial DNA in every cell. Only a single copy, one of your chromosomes comes from mom and one comes from dad in your nuclear DNA. So we have about 3 billion base pairs there that we get from both mom and dad. And mitochondrial DNA is much smaller. It's only about 16,000 bases in length, and you only inherit that from your mother. So in terms of genetic inheritance, again, you have the father's sperm and the mother's egg coming together, and the nuclear DNA is fused, but the mitochondrial DNA is coming from the mother's egg, which is why the mother is maternally sharing that DNA. So this is just to illustrate the chromosomes that you have come from your parents. And you have 22 autosomes, and one of each pair is going to come from your father. And either the father supplies either the X or the Y, and then the mother supplies one of each pair of the autosomes, the X chromosome, one of the X chromosomes, and the mitochondrial DNA. So just that's important as we talk further about genetic uses for DNA and genetic genealogy and things like that. So the current scientific thinking is that about 99.9% of our 6 billion letters that are present in DNA are the same between people. But this still means that with 0.1% difference, you can have millions of differences between people. And so if we look at those differences, we can then see the variation that can exist. So this is just another illustration where you have on top here the egg, the haploid version, or just a single copy of each of the chromosomes, autosomes here, and then the sex chromosome, X in this case, the mitochondrial DNA from mom, and then the father supplying the sperm, and again, one of each of the 22 chromosomes, an X or Y to the child, and then when the zygote is formed, it goes diploid, then you have half from mom, half from dad, and then you have the pairs of chromosomes that are now formed, and if it's a male, it'll be XY, if it's a female, it'll be XX, and then the mitochondrial DNA from mom. So if we look at the bases then for DNA profiling, we have that the genome of each individual is going to be unique. An exception to this would be identical twins, but even identical twins have minor differences, and there's been efforts to be able to try to sequence those minor differences when we do full genome sequencing to look for, can we distinguish identical twins that may have been implicated in the crime. But regardless of that, DNA is inherited from our parents, and we can use that information to be able to look across generations, which is something new you can't do with other forensic techniques in the same way. We can probe subsets for genetic variation. We're not looking at the entire DNA when we're doing DNA testing. We're just looking at a small portion of this. And this means we have to then supply statistical probabilities of a random match to this, so that we can use this to inform the stats that are applied. DNA testing has to be done in an efficient manner and reproducibly because we like those results to be able to hold up in court. Ideally, you'd like to have results that are obtained in New York City be equivalent to results that might be obtained in Washington, D.C., so that you can compare those results. And another very important point is that the DNA testing that's done right now does not look at genes. We are not looking for eye color, hair color, ancestry information, phenotypic information, or predisposal to any kind of disease. And that's really important to keep in mind. However, there are new techniques that are being developed that do allow you to do phenotyping that could look at eye color, could look at hair color, could look at ancestral, biogeographical ancestry information, and that then introduces a whole other level of complexity and sophistication that has to be considered with how is that data going to be used and who has access to it. So the markers we use in forensic testing are short tandem repeats, or STRs, and these are regions of DNA that vary by their length. And so we have, we use a technique called the polymerase chain reaction, or PCR, that copies the DNA at those target sites that we're looking at. So we're targeting STRs, we're then trying to measure the overall length of the molecule, and that then is used to then determine that size is then linked back to the number of repeats. So in this case, 7 repeats, and that would be GATA. There's 7 of those in a row. That would be then informative that that individual has what's called an allele 7. Now, those numbers can vary between people. And so you might have, for example, 7 or 8 or up to 13. And so what happens is the DNA molecule gets longer as there's more repeats in there. Well, if you just looked anywhere in the genome, two labs may get different results because one would test site A and another test site B. We like both to test site A, so we can compare them. So back in the late 90s, the FBI selected 13 core loci, core STR loci that would be run in all tests in the United States, and similar things have happened in other countries around the world. That provides a common currency. So that what that means is we can then take those results and compare them across laboratories and enable labs to be able to get results that can be shared. Now, when a test is done, I mentioned we have alleles, one from mom and one from dad, Well, you can either have what's called a homozygote, where they're both the same size. So both mom and dad, in this case, contribute a 14 repeat at this particular marker on chromosome 8, and they're the same length. So you get a single peak. If you have differences, mom and dad are different in terms of their overall length and the number of repeats, then that's called a heterozygote. And so mom contributes, in this case, a 28 allele that has 28 repeats in it, and dad contributes one that has 31.2. So the 0.2 being 2 extra nucleotides. And so there are some variations that can occur there. Now, the markers that are being used in forensic testing are these that are illustrated here. This is just an illustration of the chromosome locations. So back in 1997, the FBI defined 13 loci, as I mentioned. And we have TPOX, D3, S1358, and so on. And I'll come back and I'll explain what the name is in just a second. But we also have markers that test on the X and Y chromosomes, so we can determine the sex typing of an individual. Now, originally, with the original 13 markers, there were 8 markers that overlapped with Europe. And what that meant is we could share data, but as data sets got larger and larger, just like as you add more phone numbers in an area code, you eventually have to split that and have to add another area code or go to more digits. So instead of calling a phone number with just 7 digits, you might have to use 10 digits to make a phone call as you get more and more people in an area, or even beyond that. So now the FBI in 2017, 20 years later, moved it up to 20 loci. So now all the labs in the United States have to test at least 20 markers to put a sample into the national database. These additional markers are located on other chromosomes, like chromosome 1, chromosome 10, chromosome 22, and 19, and so on. And this now allows us to have 15 markers that are in common between the US and Europe. So international crimes can be explored more effectively. Now the naming, so you see this marker down here, D13S317. So this is similar to a mile marker that you might see on the chromosome highways, if you will, of the human genome. So instead of being route 12, mile 260.2, this would be D for DNA, 13 for chromosome 13, S is the specific sequence, and 317 is the 317th location that's been defined on this chromosome. So I mentioned other core loci, like D16 or D7, D8, D21, D18. These are all located on chromosome 16, 7, 8, 21, and 18, and so on. So I just provide this background, so whenever you see data from a DNA profile, you understand where that's coming from. It's just a marker that's located on chromosome 13, and by using that same marker, we can compare results in New York City or Maryland or California, wherever we want to be able to compare results. Because when we do DNA testing, it's important that we always have a comparison. So the DNA profile by itself is fairly useless. It's just a series of numbers, the number of repeats that exist at the sites that are being tested. So you have to compare that to a reference sample. And the ability to identify somebody only works if you have somebody to compare it to. So in the case of crime scene evidence, you're comparing that to a suspect, as we do in a forensic case. In the case of a paternity case, you'd have the child is your unknown, is your known there, and you're comparing that to the unknown, the alleged father. You may have multiple alleged fathers that you're trying to see who's the real father here. In the case of victims' remains, you're then comparing that to a biological relative. Is this person the father or the child, the daughter of the individual that's been missing? That might be in a mass disaster or so on. And also you have soldiers' remains, and again, in this case you have direct references. So the Armed Forces actually maintains a database of blood cards from everybody that serves in the military, and if somebody is killed, then they can pull that blood card and they can compare that to the remains and do that test. So just to illustrate how paternity testing is done, so here we have a mother, and I've highlighted her. This is from that marker D13S317, and she has 2 alleles there, an 8 and a 12, and then there's 3 children. If those 3 children come from the same father, you can see what would be the father's profile. Well, the 8 and 12 and the children have to come from the mother. Therefore, the 11 and the 14 would come from the father. So for this individual to be a father of these children, you would expect that person to have an 1114. So this is actually three of my oldest three children. And so I'm the father. I've been able to show with DNA testing I'm the father of my children. And I have 6 children. And what's interesting is that we used to have their profiles framed when we walked in the house. So I've taken them down now. But as they've grown up, 4 of my kids are away at college and so on. But we still have 2 at home. But it's fun kind of seeing how these genetics works. Each of them is a genetic experiment, if you will, looking at the various combinations of loci. You can see in this particular case, the combination we don't have is an 8 and 11, right? So we have the 1214, we have the 814, we have the 1112, but we don't have the 811 combination. So it was kind of a joke in my family when our kids were born. My wife would always ask, "Is this going to be an 811? You know, is that we're going to finish out this set here?" Anyway, so DNA can be used for a variety of purposes as we talk about the capabilities of DNA testing. First of all, and foremost, in a forensic investigation, the ability to identify the perpetrator is a wonderful characteristic, which with other evidence, like trace evidence, for example, you may be able to make an association, but you can't necessarily identify someone with that. We can apply a weight of evidence, and we can use established genetic principles that have been defined since 1908 for the statistics and the principles of how the inheritance happens. We can also use these established characteristics of genetic inheritance to enable close biological relatives to be used as reference points. This is important for kinship associations like we might have for paternity testing, but also for missing persons identifications and so on. Another capability is we have superb sensitivity. The ability to copy the region of DNA that we're looking at and amplify that up is very powerful. But what that also does is opens up the possibility of contamination. And so even a small amount of DNA that can be present from just touching a surface, you're leaving a little bit of DNA behind, and that can contaminate a sample that already exists there. We also have, for that very reason, there's been a lot of effort over the last 25 years into quality assurance standards and making sure that we have quality measurements or measures that are in place to help make sure we have good quality work. And then finally, we have constantly new developing technologies, and that's in large part because of genomics. The effort to sequence the human genome and to understand the value of DNA for medicine purposes has really fueled the development of new technologies, new instruments, new software, and so on. And that's led to a growth in the forensic DNA community, where other forensic disciplines don't have that same capability to draw from in the same way. But in the end, a successful interpretation of DNA requires the Q to K, the question sample, the evidence sample, in the case of a forensic case, being compared to the reference sample, the known, the K. So you have that Q to K comparison that you're always coping with. Now, when we talk about what can DNA do and what can it do, what can DNA do? Well, it can answer the question of who left the DNA. So, and again, the best results are going to happen when you have high quality, single source DNA samples. We can confirm a possible genetic relationship with biological relatives like we do in relationship testing or paternity testing. We can, with a different type of testing, we can actually assist in investigations to noting distant family relationships. That's what investigative genetic genealogy is. We'll talk about in a few moments what that can do. And then because of the high sensitivity, we can generate DNA results, unfortunately, that may not be relevant to the case because of contamination or DNA transfer. So that's important to keep in mind. Now, what DNA can't do? It can't answer the question of when or how the DNA was deposited. And so certainly the quantity of DNA can affect propositions in that regard, but it can't directly answer that question of when a DNA sample was deposited. We can't prove that someone did commit that crime with the presence of DNA, and we can't prove that someone did not commit the crime with the absence of DNA. That's important because DNA may have been removed, it may not have persisted, it may have been transferred there, and that can influence those things. So that's important to keep in mind, the cans and the cannots of DNA. So the application of DNA testing, I've already mentioned a number of these, but we have, of course, forensic cases that you may be interested in, where we're matching the suspect to the evidence. But we have lots of other uses for this, whether it's identifying the father in a paternity case, or the missing person, an investigation there, the military DNA dog tags, where they're identifying the remains of a fallen soldier. Convicted felon databases are enabling, if someone has repeated a crime, you can make connections between those crimes and then associate that back to the individual. For mass disasters, you can put the pieces back together, whether that's a plane crash, or that's the World Trade Center towers coming down, like in 9/11. And we have historical investigations, like, for example, the Tsar Nicholas II. DNA investigations were able to make some very important understandings of that. But in the end, almost all of these cases will involve STRs, the short tandem repeats we've just talked about, and then matching that to a reference sample. So one of the things that you hear about a lot in the news is the DNA database. And this is the National DNA Index System, or INDEX, that is authorized in 1994 by Congress under the DNA Identification Act. And since 1998 has been maintained by the FBI laboratory. And there's more than 200 laboratories now in the United States that provide data to that, whether it's a local DNA index system or the state. So the state police in Albany, for example, and in New York City, there's an ELDIS system there. And then at the state level in Albany, they have a ESDIS, a state DNA index system, and that data is then uploaded to the national system in order to solve crimes that may have been committed in Connecticut or Massachusetts or California or other places. And there's categories of crimes that are for inclusion based on the state law. So laboratories end up being audited against the quality assurance standards, and those have been updated over the years. The most recently was just last July. And it uses a software called CODIS, the Combined DNA Index System, to make those connections. And that software is constantly being updated as well. As we've already talked about, you have to have 20 loci, 20 STRs, to provide the common currency as kind of the baseline. And as of December, the most recent statistics, we now have almost 20 million profiles in the national database, and this has assisted more than 500,000 investigations. And so when new samples come to this is a figure from one of my books, the new offender samples are added, they're then searched against the crime profiles, or with new crime profiles are added, they're searched against the full offender database. So you're usually not searching the whole database against the whole database, just the new ones that are being added are being searched. And then new crime scene samples may be searched against other crime scene samples to get a forensic hit, or you can have an offender hit if you connect the crime scene to the overall database, to the offender database profiles. So this is what's kind of happening now. And what are some of the critical challenges that are being faced in the field? Well, I wrote about this several years ago. About 6 years ago, I wrote an article called The Future of Forensic DNA Analysis. It was published in the Philosophical Transitions of the Royal Society in London. And we had a conference there, and we talked about some of the challenges that exist in all of the various forensic disciplines. And I was the one that was asked to talk on DNA at that meeting. And really, the success of DNA testing has led to lots of backlogs. And so that means there's been a growth in submissions. The police are very excited to send samples in. In some cases, that's required that laboratories have more automation, so robotics and computer systems, that limb systems and so on, that allow tracking of data. And in some cases, laboratories have had restrictive case acceptance policies. They'll say we'll only accept 3 swabs because the law enforcement investigators may do swabathons. This is what a colleague of mine said a number of years ago, that they go in, they swab everything, and they just then submit everything to the lab. Hundreds of swabs may be submitted to try to see what they can find out, because of the success of DNA testing. The other thing that's happened is we have greater detection sensitivity. This leads to more complex mixtures, low template DNA that we have to face because of touch evidence. So many labs are introducing probabilistic genotyping to cope with this increase in the challenge of the data that they're trying to interpret. And in some cases, I think, this is my opinion, we need to implement complexity thresholds to say, well, I'm not going to go out there because this is too challenging. There's too many unknowns with this type of sample. Okay, so I want to move now and talk a little about DNA tests and methods, and describe kind of what's being done, and then we'll talk about where things are going. So if you look at the overall process, we have either a blood stain is collected from a crime scene or collected from an individual, or a buccal swab is collected from an individual. They swab the inside of someone's mouth, getting cheek cells, and that then leads to samples being collected, and they have to be stored so they can be used later. The DNA is extracted. When it's extracted, it just looks like a little tube of water. Then DNA can be quantified to see how much is present, and that will then influence the types of tests that may be done, or it can influence how much DNA you put in for the test. Then we perform multiplex Pcr, the polymerase chain reaction, which copies, amplifies, and labels the regions we want to look at. The regions we're looking at are these short tandem repeats that vary by their length and the overall number of repeats. And there are different fluorescent dyes that are attached to that. And the process of this then generates an electropherogram that's in different colors and has peaks in different colors. We can then separate those colors out with using a laser-induced fluorescence system and in capillary electrophoresis, separate out the size of the molecules. And then finally, the data is taken and interpreted, and then statistics are applied to those and a report is generated. So these are the various instruments that are used in laboratories. Most labs will use a thermocycler. We use a capillary electrophoresis instrument and then use some kind of software to do that analysis. So if we break this down, there's really two major sections. One is the measurement section, which ends in the electropherogram, what's the data obtained from that sample. And then the second part is, what does that data mean? And that's the interpretation part, saying, is this an artifact or is this a real peak that we're interested in? Could this have been arising from contamination or other things? Sometimes there's not a way to figure that out, but we try to figure those things out. So where we're moving now is on the trends that are happening here. So we're moving towards faster DNA testing capabilities and sample to answer integrated instruments, so the rapid DNA systems. We're moving towards higher information content. There's new systems, next generation sequencing systems that enable more markers and more detail of the alleles. Instead of just the overall length, we can get the sequence information from it. We have higher sensitivity. This means we have new assays and new tests that lower the detection limit. This makes interpretation more challenging. And then finally, the effort to get to stronger conclusions for mixture interpretation, particularly, are moving towards probabilistic genotyping software and doing different models for this. So we'll talk about each of these. So rapid DNA is moving into police stations. This was an article from the New York Times about 2 years ago, the DNA magic box. And it shows, this is from the FBI website, the efforts that are being made to be able to do this. So a sample will be collected when someone is booked for arrested for a crime, and in the time they're in there to be booked, and their mugshot taken and fingerprints and so on, they could do the DNA test in that time. That's the goal. The capability exists now, but how much it will be used in the future remains to be seen. And there's just more information on this slide here, just that for non-CODIS rapid DNA consideration best practices, when law enforcement does this, the FBI has a whole file that teaches you more about that. These are the instruments that are being used right now. The Andy system's on top. It can do 5 samples during a run, and then the rapid hit ID is on the bottom. It can do a single sample at a time. And these will generate results in less than 90 minutes with each sample that's tested. And will generate usually between 15 and 23 STRs, autosomal STRs, depending on which kits are being run there. And typically the success rates are around 80% or so. So they're not working every single time, but the reagents are costing about 10 times the amount of conventional testing. So it does cost more, but you can get a result much faster. And this was signed into law a few years ago in August of 2017, the Rapid DNA Act. And there's been a number of articles published with validation studies. I did a review of them about 1.5 years ago in July 2019 that I can send anyone that's maybe interested. And you can go to the FBI website and they have a lot more information on this. Okay, so if we look at next generation sequencing, we can look at additional markers, but this requires more sample preparation and there's a lot more data analysis on the back end. One of the key issues that's being worked on is how do we call those alleles? As we have all this additional sequence information, it becomes more complicated. There was an article published a couple of years ago by the International Society for Forensic Genetics, and we're in the process of have a new DNA commission working on this further. So we have STR typing has been done for more than 20 years with capillary electrophoresis. That works very well, but it just measures the length of the molecule. What's under development is further information, more markers, and getting ancestry information, separating identical twins, and other things can be done with this. But we have the challenge of how do we describe those results? How do we store large amounts of data? How do we address the privacy concerns when we're getting other information, like ancestry information and so on from those samples? So for an STR marker that's being sequenced, instead of just getting the length as being 14, this is all that would be stored in a database is that 14. Now you have the full sequence. How do you store that? How do you call that? And things like that. So this is an example of one of the markers, D12 on chromosome 12 and allele 21. If you measure it with capillary electrophoresis, you get just one peak. But if you sequence it, this is from 183 samples that were sequenced at Nist by Catherine Gettings a few years ago. There's actually 9 different sequences that can exist. And so you're getting further subdividing the information with them. Now, if we look at just a few things about next generation sequencing, or sometimes it's called massively parallel sequencing, we get these additional markers. We can get as many as 10 times as the number of markers we would normally get with capillary electrophoresis. This gives us a lot more information. We have new capabilities with phenotyping, with getting hair color or eye color or other information like that. But we also face the privacy concerns with this additional information. Requires more sample preparation, more data analysis is done, and then we have to worry about what's the cost for all of this. So it's cheaper per marker, but it actually costs more for the entire run. And then finally, it's really still in the realm of research. There's Nist and a number of others are researching this, but it's not being routinely implemented yet into forensic casework, but that could happen very soon. It's been proclaimed that it should help with mixture interpretation, but that hasn't been demonstrated yet. Next, I want to move to the impact of sensitivity. We talked about this a little bit, the contamination that can happen, but this is something I wrote in one of my books a few years ago. As the sensitivity of DNA typing improves, laboratories' ability to examine smaller samples increases. This improved sensitivity is a two-edged sword. With greater capabilities come greater responsibilities. You recognize Spider-Man in there, right? To report meaningful results. Given the possibility of DNA contamination in secondary or even tertiary transfer in some instances, does the presence of a single cell or even a few cells of an evidentiary sample truly have meaning? So for this reason, there's been a lot of work in the last couple of years on DNA transfer, what's called activity level propositions. And this is how do we cope with this increasing sensitivity? Can we give more meaning to those results? So there's a growing body of literature in this area. And one of the things that's come from this is that how little we truly understand with certainty about how DNA transfers from the donor. If someone sheds more DNA than one person may shed more than another. What are the range of variables that affect that transfer and that persistence, how long the DNA lasts on the surface and so on. The value of using activity propositions, we're trying to figure out more information from that. And then how can we do this in a more systematic way so that the data can be used by other laboratories? So the overall takeaway really from the literature is that subsource DNA results in isolation by themselves cannot automatically be assumed to relate to the crime. So what's important to keep in mind is what question are we answering when we get a DNA result? And that's something that's really important in all aspects of forensic science, is what question are you answering when you generate data? So this is from a paper that was published by Peter Gill and colleagues, a DNA commission paper that was published about 2.5 years ago, that looks at the levels of hierarchy of the propositions. So if you have a subsource sample and you're doing an investigation, the question you're answering is, who could be the source of the DNA? Under the evaluation phase, when now you have a reference sample to compare it with, is the DNA from the person of interest, the person that you're looking at? So that's the question you're trying to answer. Well, how do you go about answering that? You look at the occurrence of the DNA profile genotypes in the relevant population, and that may generate then the random match probability or other statistics you may use there. But we also have to face the variability of results. Do we have the presence or absence of alleles, assuming that the DNA came from the person of interest? On really low amounts of sample, we may not be able to identify what tissue it came from. At the source level, we're looking at, could this be the source of the biological fluid? So we get both the DNA profile and the presumptive tests that may be done for semen or saliva or other things that are being looked at. And then we have to be concerned about potential false positives or false negatives, cross-reactivities that may exist with those tests. Finally, on the activity level, the question we're trying to answer is, did the person of interest perform the given activity? In order to do this, we have to consider all these source factors, plus the DNA transfer and persistence, the recovery rate, the DNA that may be present for unknown reasons like background DNA, and this is where DNA transfer has a huge impact. Also, as we push the sensitivity, we now have to worry about sub-source. So we may be looking at only a portion of a DNA mixture. This is talked about by Duncan Taylor, wrote an article in 2008 that reviews all these propositions and the different things, the different challenges that are being faced there. Well, let me take an example from a recent paper. This was published last September, and this was a paper on DNA transfer to firearms in alternative realistic handling situations. So what they did is they examined DNA mixtures from the skin contact traces of DNA that were recovered from 3 different portions of 2 different firearms. These were handled in 4 different realistic casework relevant handling situations. So someone handles the gun, they may be taken away from them, and someone else handles it. Can you see both people on the gun and things like that? Because this is where a lot of testing is happening now, is on the swabbing of guns. So how much DNA is there in the background? So from this, they looked at this, and you can see the legend here on the right. The owner's DNA, in the first particular scenario that they tested, only the owner's DNA was there. So the owner handled the gun for 4 days, touched the gun, and then put it back in a bag and so on. Then they came to a site and they pretended to shoot the gun. They went through the actions what they would do to fire the gun. The gun was then swabbed and DNA profile was generated. And sure enough, as you'd expect, the owner's DNA was there more than 70 to 80% of the time. However, there's unknown DNA that was present, and that could be from someone that shook hands with the owner and their DNA is transferred to the gun, even though they never handled the gun. Now, if you have a second handler, a shooter is illustrated here with the second bar. So in this case, if the shooter only handles the gun for a short period of time, a few seconds, and then their DNA swab, in this case, it comes up. There's DNA there, but you also see the owner, and you also see some background DNA from someone that's not been part of the test. Then if the second shooter, the second person handling the gun handles it longer, sure enough, you get more DNA there, but you still have to deal with these backgrounds that are present. And then finally, the last one was the second person then wipes the gun down after they've handled it, and you can still see their DNA there. You can see the original owner, and you can see some background DNA. So the point is, there's background DNA that's there, and so what you're detecting is very important to appreciate. Is it the person that may or may not have handled that gun? So there's a lot that we're learning about DNA transfer and persistence, and this slide just summarizes some of the things that have been learned there and some of the review articles that have more information on the promotion of activity propositions and the effort that goes into generating those, on mechanisms for how DNA is transferred from donors, a very comprehensive review of DNA transfer as of 2 years ago, and then the scheme for a more systematic approach to data collection. But the overall takeaway is that when you answer the subsource question, you can't automatically assume that they're connected to the crime, just like I showed in that last slide with the firearm being handled by somebody else, and there's background DNA on there. Okay, so as we get to more and more sensitive techniques, we really have to face the fact that DNA evidence is becoming more challenging. So I like to use a math analogy to explain that. So we have, with arithmetic, you can think of basic DNA collecting for databases like basic arithmetic, 2 + 2 = 4. Do you see those 2 alleles or not? Then when we get to sexual assault evidence, there's a lot of DNA there in many cases, and you usually know the victim. Then can you figure out the perpetrator? You can do algebra and subtract that out. But when we're doing touch evidence samples, we're really doing calculus, and it's more complex. And so you're dealing with these more complex mixtures. How do you cope with this? So in the real world, data that you're facing in a forensic laboratory, you have the true heterozygote, you detect both alleles, but there's stochastic or random variation that happens when you're sampling low amounts of DNA. And the PCR process is affected by both the quality and the quantity of DNA. And so this imbalance gets worse when you have low amounts of DNA. So you may have allele dropout. And then you get a false homozygote. You only see a single peak when there should be two. And that second one is not there. And that can also happen because the DNA is degraded or because you have PCR inhibitors that prevent the amplification, chemicals that prevent the amplification there. And then for mixtures, you have to deal with overlapping alleles. So it may be challenging to determine, is this a stutter artifact that exists or is it a true allele? And so you have to consider all those possibilities. And this is where probabilistic genotyping software is coming in and being used now. There's two primary approaches. One that just evaluates the alleles that are present with the probability of dropout. And in New York City for many years, they've had the FST, the forensic statistical tool. Lab retriever has been used by several labs out west. And then there's the continuous approaches that utilize both the alleles and their peak heights. And this is two software programs that use this are StarMix and TrueAllele. And they use statistical modeling to go through and calculate the probabilities of different genotype combinations being present. And then they use those in conjunction with propositions like the number of contributors that are there and calculate likelihood ratios. So there's multiple programs that do this, multiple models, and there's still a lot that's being learned about how these models are performing, how they perform with different types of samples. Some are open source, some are proprietary. And so this is something that's really a growth area, what's happening in the field right now. So there's several review articles I would recommend if you want to learn more about this. They're both were published in the January 2019, so about 2 years ago. DNA Transfer, which has almost 300 articles about DNA transfer. And then this bottom one on probabilistic genotyping software and overview that provides a nice overview of where the state of the field was 2 years ago. So I want to finish with talking a little about genetic genealogy. This is something that you read about in the news. In fact, it was in the Atlantic last Wednesday. So a prediction I made a couple years ago in writing one of my books at the end of one of the chapters was the future may involve more interaction between the application of genetic genealogy and forensic science to help bring cases to closure without prior suspects. The reason being because there's lots of data in these databases, and if we can use the ability of various markers to extend beyond close relationships, we may be able to learn more. So there's a lot going on in this field right now. Ever since the rest of at that time, the alleged Golden State Killer, he's since been convicted. I'll show in just a moment. But in April of 2018 now, almost 3 years ago, this opened the floodgates to this. And it's been used to solve a number of cold cases and with the non-law enforcement databases. But this has also raised genetic privacy concerns. And it's been a very, many meetings I've been at in recent years, there's been a set of active discussions on this topic. So I include several articles in here. And there's also a link to a blog I wrote about 2 years ago on this, kind of what's happening in this field and kind of a little of the basis for how it works. Even in 2019, Barbara Ray Venter, who was the one involved in capturing the Golden State Killer, made the Time 100 most influential people. So in the Golden State Killer case, more than 13 murders had been committed, more than 50 rapes, and more than 120 burglaries that were linked by DNA evidence and other aspects of forensic investigations. And finally, a crime scene sample, a sexual assault kit, was examined with genetic genealogy markers. And that data was then uploaded to a genetic genealogy database called GEDmatch. And that located potential relatives for this perpetrator. And then a lot of work was done, creating family trees with possible links and lots of detective work, hundreds of hours of detective work to narrow down to potential suspects that then led to a confirmation sample being collected from the door handle of a car and from a tissue left in the trash, in the garbage, that then showed that this was the suspect's DNA profile was the same as the crime scene profile. And then Joseph D'Angelo, age 72 at the time, was arrested out at his home near Sacramento, California, and he was just sentenced to life in prison without parole last August. So this is new developments there. So a lot's been going on in this area in the last couple of years, and this is something that you'll certainly hear about in the news. The FBI, Tom Callahan from the FBI, published an article in Science about a year and a half ago now called Responsible Genetic Genealogy, and the FBI actually and the Department of Justice actually adopted a policy on how forensic genetic genealogy would be done. And a lot of efforts went into making sure that good practices would be done there. So more than 100 cold cases have been solved with genetic genealogy since the Golden State Killer, and you'll see those regularly in the news. And even back in 2018, there was a survey done and published in PLOS Biology, should police have access to genetic genealogy databases? And at that time, 91% said yes of the general public that were right after the Golden State Killer. There's been more discussion about this. In The Atlantic, about a year and a half ago, published The Messy Consequences of the Golden State Killer Case. Susan Zhang wrote an article about that. And then more recently, just last Wednesday, Susan Zhang and a colleague wrote this article. I want to talk just briefly about it. So this is called The Victims Left Behind by Genetic Genealogy, published January 27th. And I just take some quotes from this article. I invite you to read the whole thing for yourself. But genetic genealogy cases require investments of time and resources and often involve hiring outside genetic consultants, like Barbara Ray Venter was hired to help with the Golden State Killer. So far, this new technique has been applied primarily to cases with white victims, reflecting biases in the criminal justice system and in society at large. Our analysis found 104 murder victims whose alleged killers have been identified through genetic genealogy. Of the 89 murder victims whose race investigators shared with us, only 4 were black, 79 were white. And then they have a link to things. The 3 criminal justice experts separately suggested that detectives and criminal investigators, more than 80% of whom are white, might unconsciously gravitate to cases with victims of the same race. So you can think, are they playing some kind of race card with what they're doing? That's what the authors are trying to get you to think. Our analysis also found that homicide suspects identified through genetic genealogy are more likely to be white and less likely to be black compared to national statistics. Homicide victims and offenders are usually of the same race. Starting with more white victims is likely to lead to more white suspects. And then this one. This is almost at the end of the article, but this is really the crux of the whole thing. C.C. Moore, who's a genetic genealogist at Parabon, says black suspects and victims are harder to identify with genetic genealogy since the existing customer databases skew towards people of predominantly European ancestry. So the reason that more white victims or more white suspects are being connected is because there's more white people in the databases. And so the genetic genealogy databases, it's not for some predisposed, in my opinion, racial predisposition of detectives or anything else. And so you have to be very careful when you read these things. So I bring you back to the quote from Cordelia Dean. Activists spin it, journalists misinterpret it or hype it. And I believe that's what happened in the article from last week. So be careful how you interpret what you read, but the point is there are limitations with any technique, and there's great capabilities with DNA, but it won't solve everything. So with that, I'll just leave you my contact information. Say thanks for your attention and be happy to take questions as we go onto these things.